Does this work? Next. Sorry. Thanks to the organizers for this wonderful opportunity to come here to this lovely campus. It's great to come here, actually. It's a fantastic institute. And today I'll be speaking on counting RNA structures. And this is work done with my student, Pradeep Badola, in the Department of Physics, Delhi University. Here's a little introduction to RNA. RNA are biomolecules which transform information from the information storage biomolecule DNA. Through translation, form proteins which form the biological function. There are three major types of RNA, RNA, mRNA, and tRNA of these particular lengths. NT means the number of nucleotides present. The chemical composition that of the nucleotides are the usual ACGC, and they are connected by the hydrogen bonds through G with C, A with U by the Watson prick, and the wobble is with GU, and these are the energies with which they interact. So the hydrogen bonding is responsible for secondary structures in the RNA, and in the DNA, of course, they're responsible for the helical structures. This is a picture of the RNA with its sugar phosphate background, where the nucleotides are positioned this way. There are three levels of structure that are found in the RNA. They are very distinct. This is the primary structure in which you have the nucleotides or the bases in a string, and the secondary structures in which they bond through the hydrogen bonds in this way, and tertiary structures in which there are the bondings are in this particular fashion. And this bonding are controlled by the electrostatic magnesium ions. So in this talk, what I will try to do is do an analysis of the database and show you some results in this database. And then I will try to give you a model which will explain some of the results that I will present, not all of them. So there are two databases that I look at, the pseudobase and the RCSP protein bank data. And these are data banks which contain all of the RNA known. So we process these, well, my student processes these PDB files, and this is the PDB file, and one views this PDB file by the RNA view, which arranges all the hydrogen bondings in the interaction, including the sugar-sugar interactions, and then you process it, keeping only the Watson crick and the wobble base pairings, and you get a graphical edge list format from which you can calculate the genus. Here's a snapshot of the processed files. This is all the hydrogen bondings in intermediate step in the processing. And then this is the graph edge list format. And then one puts it in the circular format. Okay. So this is a representation of the RNA in the circular picture. And one can calculate the genus of this graph of RNA, and it's the genus 1. These are several different representations of RNA. This is the linear representation, this is the circular representation, and this is the 2D representation of an RNA molecule. Now, one can calculate the genus of an RNA graph, and the genus G is defined to be the number of closed surfaces, the closed surface that can be drawn, that the RNA can be put in with in which the lines do not cross. Okay, so I've just demonstrated here. This is sort of a sphere, if you can imagine this. The RNA, if it is a structure which is of this one, okay, these are the RNA. Okay, so here is the RNA structure which I've just wrapped around on the surface. You see that these two lines are not crossing, and if that was all that the RNA would be, then it would be a genus 0 surface. But because of this line crossing, I have to have something else so that it doesn't cross, and that's why I have to go to the genus 1 surface so that it doesn't cross. Okay? So I have to put it around this handle here, and hence the genus, the surface of the genus for this graph, the RNA, which has this diagram, okay, in the circular representation, that has genus 1. So one can actually classify using this definition. One can classify, this is the definition, what the genus of all of the RNA graphs are. Okay. Obviously, doing this to real RNAs is a very difficult problem. Okay. So I'll be describing, I hope I'll have time to describe the model, field theoretic random matrix model, which actually enables us to do this counting in an easier way. And that involves writing the linear graphs that I had shown you in terms of double lines. Okay, so there are Feynman diagrams, Feynman rules that are present in the field theoretic model that I will describe later, and that will enable us to calculate the genus easily. If I make the graphs that I have found in these databases, okay, so I can calculate the genus, okay, using the Feynman rules described by the field theoretic model that I'll describe later. Okay, so here is an example of some of the rules that I have to use in order to find out the genus. Now, this is one result that we've got using the data. This is the number of base pairs versus the length, okay, for RNA-Riboswitches, and these are 130 PDB structures. Okay? So here is the number of, okay, for this length, these are the number of base pairs that you'll get, okay, and so on. And we notice that if you just have the wobble, okay, the Watson-Crick and the wobble base pairing only, the slope is about 0.37, and if you can consider other interactions, it becomes, of course, 0.49. Now, this is another result, which is that you're counting the number of normalized structures that you have in this database, and this is with length, okay, and then you see a peak here with size 0.3 something, and at length about 75. Here, at a length of 75, we have represented the structures in this bracket notation, so-called bracket notation. At length 75, the frequency of there are only 17 structures that the RNA takes at length 75, and these are the category of 6, 2, 3, 4, 5, 6 structures, and they appear, the frequencies are 4 to 3, 6, 1, 1, with minimum free energies of these values. These are in joules per mole and with a negative sign. Okay, the most minimum one is this 37.8, but there's only 2 of them. Okay, it looks like the least, the most abundant occurrence of structure is not at the least minimum energy. Okay, so these are the different structures. These are the different structures that this group of 75 RNA take. So here is the same group of RNA of length 75, and this is the plot, the frequency versus the minimum energy, and you see that the maximum number is around at minimum energy minus 3.2, and but if you take a random generated RNA sequences and you take 1,000 of them, this is the distribution. So you can compare these, and you see that all of them, there are gaps in what we have in our databases. Now, here is a picture of how the bases are paired, the pairing probabilities of the real RNA of these 75. And it is compared with the random sequence of 1,000 RNA. Okay. So this is comparison. And this is for a particular family, okay, in which we've taken 61 sequences. So in this family, there are 61 sequences, and they're of length 52. And we've also figured out this frequency versus minimum free energy graph and compared it with the random 1,000 sequences. And this is again the pairing between pairing probabilities within the family between the various nucleotides. Okay, and this is compared with the random. This is another family, and you see the structures. So this is about this family with 51 sequences and of length 54, compared with 1,000 randomly generated RNA sequences. And this is again the pairing probabilities for this family, and you see the structure. And here, what we've done is taken the real RNA data and using the program RNA heat, okay, calculated for different lengths, 30, 40, 70, 90, 120, 161, the specific heat versus temperature curve. And we find that that there are bumps in the specific heat curve. And this is the model. That we'll try to fit some of the data. So this is a field theoretic random matrix model, and the idea is that at each nucleotide you put a random matrix, and I stands for where the position of the base. AL is normalization constant given by this. Phi i's are the random Hermitian matrices at each nucleotide, and Vij is the interaction matrix between the nucleotides. So this Vi. This model was proposed by Henry Oland and Z, and further work was done by Vernizzi Henry and Henry Orland and Z and other people.
So your time is getting over.
Yeah, so this is the model. And what we did is to introduce this interaction, and it's basically a logarithmic interaction, which is called the PINA interaction. Work is done in this thing, in these pieces of work. And making some simplifications, that is the interaction strength being V, taking them all to be the same, the external interaction being W, and taking W equal to N over 2, we can do the following analysis, and we find asymptotic results for the partition function to change from the Orland Z value, which was L to the 3G minus 3.5 to this one. So we found another exponent here, which is a very important exponent in this area. And from here, we've calculated the genus distribution function, which are the normalized structures versus the length at fixed genus. Okay. So this is a comparison between the model that we have and the model that Orland and Z had. And the important point is the experimental genus 1 curve that we saw, experimental means from the data, is this one. And one can, because one has a parameter, one can dial the parameter to fix the correct height of the this. The second point is that the specific heat that we found from the specific heat curves from the data analysis are very, the bumps at least are very well described by this model and for various lengths. And we also find from this model why the bumps are present, because if we calculate the specific heat for the planar structures, we see that the bumps are there. But if we do the specific heat calculations,
I think the time is over actually. So would you please conclude? Or?
Yeah, if you do the specific heat calculations for the higher genus structures, these bumps disappear. Thank you, and I concluded. Sorry for your.
Any questions? If there are no questions, then please thank that speaker again and let us invite the next speaker.